Hello, my name is Saad Usmani. I'm the Chief of the Myeloma Service in Memorial Sloan Kettering Cancer Center, and I'm here at ASH in sunny San Diego. It's been a good meeting. We had a lot of data, updates on clinical trials, some new data as well. We had updates on the CARTITUDE-4 data set, specifically in context of MRD negativity and sustained responses and how the PFS is panning out for different disease subsets, and it appears to be looking pretty decent. I'm more interested in the high risk and those early relapse patients, and it appears that we are seeing durability of response in those subsets. We do have several options in myeloma in that space, but I think amongst the options that are currently FDA approved, I think ciltacabtagene probably is the best one, especially in the indication that they have in the first relapse setting. For patients who are relapsing within the first 2 years of being diagnosed or have high risk disease, I think it's a really good option. The other ones that I'm really excited about and they're coming along are the Dream 7 and Dream 8 studies. So these are the triplet combinations of belantamab mafodotin with bortezomib dexamethasone and with pomalidomide dexamethasone. And so as we get more follow up on those data sets, I would be really interested in looking at what's happening to the high risk patients. And those actually may be very good BCMA-directed options for that high-risk subset. I think you can make a case. Certainly, we're exploring that question in the newly diagnosed setting. We can certainly explore that question in the highest risk smoldering myeloma patients where we can get a really nice intervention going. We also have to pay attention to not just the risk of progressing to myeloma, but also disease biology. So there are going to be certain subsets where, I would say, yeah, we can use these kind of immunotherapies. Is it something that I want to employ for high-risk MGUS or low-risk and intermediate-risk smoldering myeloma? That's where the risk versus benefit proposition is very different. So we have to be very careful in what we are trying to do for those patients and not cause harm. I don't think that you have to, there's a saying, you have to worry about the first cancer first. And, it doesn't look like there is a huge incidence. So the incidence is fairly low. It's in single digits. And for certain types of SPMs, it's actually in low decimal points. So we have to be a little bit careful in interpreting those data. However, more important are the infection risk, the cytokine release syndrome, and potentially neurologic side effects that patients can have. Some can be residual, and that can impact the quality of life. So again, it's the risk versus benefit ratio. And that's why I was making the comment that when we talk about asymptomatic precursor disease, any intervention we pick, we need to make sure that we are not compromising the quality of life and giving patients side effects that they otherwise shouldn't have had. I do feel that for standard risk patients, we have a lot of options, and that's where I think sequencing of therapy would make more sense. We can probably look at CAR eligible versus CAR ineligible populations, or CAR accessible versus CAR inaccessible population areas, and pick the right treatments. I do feel that for high risk patients, it will be a combination approach that gets us to the finish line. We'll see some real world evidence being generated with standard of care patients. There are also clinical trials looking at that sequencing question. And so these will be smaller experiments or smaller clinical trials where either you are using it as a bridging strategy. When I say it, I'm talking about bispecific antibodies, or you're using bispecifics right after the CAR T cell therapy for a short duration of period to deepen the responses and get better progression-free survival for patients. So I think we learn, this is a very active area. What was the most fun thing for me to see at this ASH was the number of younger junior investigators interested in the field and coming out and presenting these data. So I think we are going to have a lot of robust data available over the next few years, and it will help guide which direction we want to take for different subsets of patients.